Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Illuminating Pathways for Disease Research. The complexity of biology continues to challenge the ability of researchers to find answers through the application of various omics techniques. While genomics, transcriptomics, proteomics, and metabolomics are widely used in both industry and academia, these experiments generate a huge number of measurements. Also, there are times when a single omics technology does not provide enough signal for a statistically valid identification of a discrete set of target entities to be made. However, integration of data from multiple omics approaches can provide enough constraints to greatly reduce the false discovery rate. This will be the focus of today's webinar, and we've assembled an expert panel to explore this topic. Let's meet the panel. Dr. Q. Rhee is an Associate Professor of Medicine, Microbiology, and Immunology in the Division of Infectious Diseases at the Weill Cornell Medical College. Q is going to discuss how an integrated systems approach can help elucidate how antibiotics work. Dr. Benedict Kessler, University Research Lecturer in the Ubiquitin Proteolysis Group at the University of Oxford in the UK, will explore translational proteomics and metabolomics using a case study on ankylosing spondylitis. And Dr. Akhilesh Pandey, he serves as a professor at the Johns Hopkins Bloomberg School of Medicine. He carries out research at the McKusick-Nathans Institute of Genetic Medicine in the departments of biological chemistry and oncology. Akhilesh will describe the results of an integrated transcriptomic and proteomic analysis for determining the pharmacodynamic action of sulforafane. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So if everyone's ready, let's get going. Dr. Q. Rhee will be our lead-off presenter. Q?
Thank you, John. So what I'd like to talk about today briefly is at least the approach my laboratory has taken to integrating different types of system-level measurements to better understand how antibiotics work, with the ultimate hope that this type of approach will more efficiently improve the antibiotic development process. So next slide. Just by way of background, my laboratory is interested in tuberculosis because for a couple different reasons. Because it's a disease that lies at the intersection of what we think are two critical public health crises. On the one hand, tuberculosis is the leading bacterial cause of death worldwide. It afflicts 8 million people each year and claims the lives of approximately 2 million. And what's more is that over 1/3 of the world's population is actually infected and at risk for developing disease over their lifetime. And therefore, it's a disease of global proportion, and it has social implications because it's actually a socially predatory disease that actually disproportionately afflicts the most resource-poor populations of the world. And this is actually the primary motivating factor for why we study tuberculosis. But in addition to studying it from the standpoint of the global health emergency, we're interested in it also because, next slide, we believe it exemplifies an equally significant but less well-recognized public health crisis, and that is the general and increasing failure of anti-infectives research to produce virtually any new classes of antibiotics in the past 40 years. So it's widely recognized that, of course, that new infectious agents emerge and old ones reemerge, and antibiotic resistance seems to be almost the inevitable fate of every antibiotic in clinical use. Yet, despite the fact that microbes evolve so fast, our ability to develop new antibiotics has increasingly fallen behind, such that despite the fact that infectious diseases and anti-infectives research were one of the earliest triumphs of modern medicine, infectious diseases has paradoxically become probably the only area of medicine to become steadily less and less effective over time. Next slide. And so the reasons for this shortfall in antibiotic development, or the so-called problem of bad bugs, no drugs, is clearly multifactorial, and a lot of attention has been paid to the economic factors that have driven this kind of drought of antibiotic development. However, there are also several key underappreciated scientific shortfalls that we think have largely gone unnoticed. But represent real scientific challenges to developing anti-infectives and helps explain why this has become such a brewing public health crisis. And so some of them are outlined on this slide. So probably the primary reason is that, in fact, if one takes a hard look at the history of anti-infectives development, it turns out that the success of anti-infectives and how it's been approached has really been an empirical and not a rational-based approach, meaning that it's largely trial and error and a fair amount of luck. And as a result of this, because we've been taking this kind of empiric approach and had success with empirical approaches to discovering and developing antibiotics, it's very difficult to predict the development of new antibiotics because we really have very few, if any, examples of antibiotics that have been developed along a rational framework rather than an empiric one. And a corollary of this is that for many cases, we actually lack an understanding of how the antibiotics we have actually work. So what I mean to say is that we understand how many antibiotics act on a biochemical level, but we don't understand why the activity of a given antibiotic against a given protein target, for example, ultimately leads to growth inhibition or killing, or how an antibiotic works. And in practical terms, what this has amounted to is that we've really been unable to, because we've never really rationally been able to successfully develop an antibiotic. This really reflects the fact that anti-infectives research has historically been dissociated from the power of kind of modern 21st century science in the era of genomics, structural biology, and medicinal chemistry haven't really been brought to bear to their full potential because of the fact that these sciences are really rationally driven, yet our approach to antibiotic development has largely been empirical. And in maybe slightly more practical terms, we think that antibiotics, of course, act on the cellular biochemistry, but the tools that we have to study them have really been non-biochemical in nature and inferred, so we rely on the use of indirect readouts. And the way this is important is that from the standpoint of kind of modern rational drug discovery, the approaches that we have now largely lack some key aspects of drug activity that we'd like to know, and these involve chemistry, stoichiometry, and kinetics. So these are typical things that are not encoded by traditional genomic-based approaches. And so we think there are some real key scientific shortfalls in our ability to translate biological information into pharmacological applications. So next slide. So as a solution to this, we've taken the view that we'd like to adopt metabolomics-based approaches as a bridging tool that will allow us to basically bridge the translational gap between biological information that's provided by genomic approaches to medicinal chemistry. And the main reason for doing this is that, in our view, metabolomics offers systems-level readout of a biological system in biochemical terms. So to the extent that drugs act on cell biochemistry, we think that metabolomics probably provides the phenotypically or pharmacologically most useful form of information to rationally guide drug development. However, that said, there are also inherent limitations that we faced in trying to develop metabolomics as kind of a standalone approach to drug development, and these are listed as so-called cons. And one, of course, is that metabolomics measures metabolites, but not all drugs may act on metabolic targets, and that's almost certainly true to be the case. In addition, all biological systems and phenotypes involve the operation of multiple interacting networks that involve genes, proteins, and metabolites, so that any one approach may not provide enough information to really understand how a given compound acts on a biological system, or in this case, microbe. And the third is probably a less recognized limitation of metabolomics in that compared to other omic type of technologies, metabolomics is inherently analytically challenged by the fact that the chemical diversity of metabolites is much larger and greater than that of proteins or nucleic acids, and what this means is that the amount of coverage is inherently incomplete because there is no single analytical method, at least today, that can cover or embrace the chemical space of all biological molecules. So unlike other omic approaches, the use of metabolomics, while it provides a biochemical readout, it doesn't provide the same type of complete coverage that genomic or even proteomic approaches can offer. So Nonetheless, the fact that metabolomics offers those three key properties that I discussed previously that we think are important to drug discovery, chemistry, stoichiometry, and kinetics, we think that the qualitative nature of having a biochemical readout from an intact cell is actually highly desirable. And so in weighing the pros and cons, we were interested in trying to find a way to further augment or enhance metabolomics-based tools to enrich the current drug discovery toolbox. So, next slide. The hypothesis for our work is that we came to the realization that perhaps we could overcome some of these analytical as well as conceptual limitations that we just discussed about metabolomic approaches by now merging different types of systems-level data sets, and in particular, metabolomic and transcriptomic data, and that by merging the two, we might be able to better understand the mode of actions of existing as well as newly discovered antimicrobial compounds, as well as to facilitate the process of identifying the targets of new compounds that we don't understand or old compounds that we have but still don't fully understand. Next slide. So to do this, we collaborated with Agilent to guide the development of the so-called Mass Profiler Pro / GeneSpring multi-omic software analysis tool as both a practical as well as conceptual means of merging metabolomic and transcriptomic data sets so that in the single software package we'll be able to co-manage both types of data sets using the existing and already proven power of GeneSpring-based platforms and analytical tools, and then to not only process the primary data within the same software package, but then to be able to, next slide, directly visualize the integrated data sets by projecting them onto biological pathway diagrams. And in particular, what we wanted to do was we wanted to be able to have the flexibility to project our data sets onto biological pathways that are specific to our system, and so that we're not constrained to using already either model or already fixed pathways that are already out there, because this, of course, biases against our ability to learn things that are truly organism-specific. So we wanted to maximize the specificity of our data set analysis for the system of interest. And so we coupled these GeneSpring tools to the use of pathway tools using the WikiPathways architecture, which is an open source-based pathway visualization tool that allows one to construct and curate custom biological pathways from the system of interest. And in this case, we've done so for Mycobacterium tuberculosis, the agent that we're interested in, and then projecting the data sets that we're able to generate onto these custom pathways to really try to maximally enrich the amount of biological or system-specific information we could extract. Next slide. The case study that we applied this to were for a class of antibiotics that are in wide clinical use because they're often considered first-line TB drugs. They're highly active, and it's a case where we understand what the primary target of the antibiotic is, yet we don't fully understand its mode of action. So aminoglycosides are protein synthesis inhibitors that are known to bind the 30S subunit of the ribosome and corrupt the ribosome function to cause mistranslation or premature termination of protein. But how this ultimately leads to cell death is unknown. Moreover, we know that this target is not a metabolic enzyme, so we thought that this would be a good test of how informative emerging transcriptomic and metabolomic data sets onto pathway visualization tool would be in expanding our understanding of how aminoglycosides work, and whether or not, in fact, for a drug that doesn't have metabolic enzyme as a target, whether the metabolic signatures are actually informative of their mode of action. And this, we think, holds practical importance both from the standpoint of being able to deduce the target of a compound that may act through a similar mode of action but be structurally unrelated to aminoglycosides, for example. So next slide. So to start, we generated experimentally matched metabolomic and transcriptomic data sets for MTB treated with a standardized dose range of 3 different antibiotics, 2 aminoglycosides, amikacin and streptomycin, and a non-aminoglycoside, ethambutol, that targets a metabolic enzyme involved in cell wall biosynthesis. And so shown here, both principal components analysis, and I guess not shown here is a hierarchical clustering analysis of their metabolomic profiles, which in this case we limited to only named compounds because we didn't want the overall profiles to be influenced by the presence of the drug or their biotransformation products. We could observe that, in fact, their metabolomic patterns showed a clear dose-dependent segregation of aminoglycosides and their ability to discriminate aminoglycosides from non-aminoglycosides is shown by the fact that the net vectoral direction of movement of the metabolomes of amikacin and streptomycin, the 2 aminoglycosides, are more closely related to one another and distinct from that of ethambutol. So this analysis was highly encouraging because, of course, it indicated that while the primary target of the aminoglycosides themselves are not metabolic enzymes, it's the ribosome, their activity was nonetheless reflected in their metabolomic profiles, which to us indicated that ultimately the biochemical action, in fact, is ultimately has an impact, has specific effects on the underlying metabolic network. However, by the same token, because aminoglycosides aren't metabolic inhibitors, aren't inhibitors of known metabolic enzymes, what was less clear was whether or not to use these patterns for any more than their discriminatory power to say that aminoglycosides look different than non-aminoglycosides. And in fact, when we tried to just map the primary data sets on the pathways, many of these pathways were only sparsely populated because the number of metabolites represented per pathway was relatively sparse compared to the number of components in that pathway. Nonetheless, this initial profiling convinced us that, in fact, the metabolomic profiles had discriminatory power and could be used as a biochemical discriminatory power for the activities of different antibiotics. Next slide. Now, the transcriptomic profiles, which have been well studied and in our hands recapitulated the same thing, also showed a similar type of discriminatory power in that, again, we could see that streptomycin and amikacin, the 2 aminoglycosides, the dose-dependent effects, were more closely related to one another than they were to ethambutol that moved in a different direction. However, like the case for the metabolomic profiles, we also face the challenge of not being able to readily interpret these patterns solely based on their annotations because there's an inherent uncertainty regarding the accuracy, completeness, and faithfulness of these transcriptomic profiles to the actual biochemical effects of the antibiotics. So that I think it's true in most microbes, it's certainly true in TB and probably other organisms, that in fact only 40% of the annotated genome actually bears homology to genes of known function, and in fact there are equal number of genes that don't bear homology to known proteins of known function. And so this leaves large dependence on the accuracy and completeness of bioinformatic annotations in terms of being able to interpret them, notwithstanding the potential for impact of annotation errors. Next slide. So as a test of this, what we did was we then merged the metabolomic and transcriptomic data sets to identify pathways for which metabolites and genes were represented, and then overlaid the 2 sets of lists, pathway lists, one for metabolite-scored pathways and the other for gene-scored pathways, to identify common or overlapping pathway patterns that were seen across both data sets. Next slide. And so, in fact, this pathway analysis of both genes and metabolites revealed a strong and what seems to be specific enrichment for some but not all amino acid biosynthetic pathways. And so as an example, we show here the results of the gene and metabolite hits for the cysteine-methionine biosynthetic pathway. And so here we could see that there's a clear enrichment of metabolites in this pathway affected by the aminoglycoside, and that this identified this pathway as a potential area or region of metabolic reactions to look at where we might be able to transform the discriminatory power of the metabolomic data set into mechanistically or biologically useful information about the activity of aminoglycosides. Moreover, in addition to just grossly scoring to say that a particular pathway was enriched for aminoglycoside induce changes in gene transcript and metabolite levels, we could actually use the same data set, having flagged the pathway, to more precisely localize specific pathways and reactions that were affected within this relatively large pathway by aminoglycosides by recursively analyzing the same samples and data sets for additional metabolites that we didn't detect using our initial analytical and computational survey methods. So again, this comes back to a very practical issue of coverage of metabolomic data sets in that they're inherently incomplete owing to the chemically diverse nature of metabolites compared to other molecules. Nonetheless, if we could identify a pathway, we could then go back and remine either samples or data sets to look to see whether or not, in fact, we can more richly populate this pathway for affected intermediates to more specifically localize subsegments or regions of this pathway that are affected. And so here we focus specifically on cysteine methionine just based on the absolute enrichment of both genes and metabolites affected by aminoglycosides here. And then we were able to go back, next slide, and recursively remine the same data set for additional metabolites in this pathway. And what we could discover then was that there was actually, we could begin to discern a specific pattern of changes in intermediates, such that the ones circled in green where it went down, the one that's in red went up, and the three that are in black were unchanged. And the ones with the dotted lines were additional metabolites that we didn't pick up initially, but we were able to discover upon remining just the same data set without doing any additional work, and found that, in fact, the way it looks like was that when one put together all the changes, it looked like one action of what aminoglycosides was doing was to cause a redirection of intermediates involved in methionine-related metabolites to maintain their levels. And so methionine, of course, is an essential amino acid that in bacteria is required for the synthesis and translation initiation of all proteins, but is also an essential cofactor in the synthesis of the ubiquitous methyl donor S-adenosylmethionine that's required for many essential methylation reactions in bacteria. So pointing us to the notion that, in fact, aminoglycosides, among their potentially many actions, one specific action may, in fact, be to impose a form of methionine oxytrophy or stress on the cells such that they redirect metabolism to try to maintain normal levels of these intermediates, and that perhaps their killing may, in fact, in part be explained by a dose or exposure that overwhelms the cell's ability to maintain levels of N-formal methionine, and or S-adenosylmethionine. Next slide. And as an additional validation of the idea that, in fact, that these metabolites may be limiting, was that when we looked across the microarray data set, we found that looking at functional classes, probably the single most enriched class of genes that were affected were those involved in SAM-dependent methylation reactions, particularly those involving the ribosomal components. As a contrasting example, we also found that this type of pathway projection could also, the gene alterations in gene levels could also be used to direct our attention to specific pathways, because there were some pathways for which there were a disproportionately large number of genes afflicted, but we didn't see very many metabolites. And so by flagging our attention to these pathways, we could then go back and begin to remine the data for additional metabolomic coverage as evidence to identify additional potential biochemical targets affected by the aminoglycosides that weren't detected by our initial metabolomic profiling, again emphasizing how this type of pathway enrichment approach can help deal with the analytical limitations in coverage offered by metabolomics, which again is kind of a unique distinction from other type of omic technologies. So next slide. So in summary, I think this short case study helps to illustrate the both conceptual as well as practical value of being able to integrate metabolomic and transcriptomic databases to not only improve the coverage of the shortfalls in metabolomic coverage offered by most methods, but to actually enrich our biological understanding of how, in this case, how antibiotics may work and facilitate target identification, and from a broader perspective, how we can go beyond taking these large data sets and translating their discriminatory power into biologically mechanistic information. So our next steps are now to then pursue orthogonal experimental approaches to define and or validate how functionally important, for example, these changes in methionine are to determining the potency of aminoglycosides, for instance. So in closing, next slide, I'd just like to acknowledge a large and wonderful group of collaborators I've had the privilege of working with and teaching me about so many different aspects of the science, ranging from the clinical medicine all the way to the mass spectrometry that involves actually a rather unusual collaboration between academics at different institutions, as well as the involvement of industry scientists, including those especially at Agilent who helped us champion and pioneer this, or gave us the privilege of co-developing this software with them, or at least to guide it. We probably didn't do so much the development so much as we provided advice. And so with that, I'll turn it back to you, John. Thank you.
And Q, thank you for that very incisive talk on integrated systems and their application to the study of antibiotics. You raised some very interesting points, and I'm sure that members of our audience are going to have questions for you. So thank you again. For those of you who have just signed into our webinar, we're delighted that you've joined us. We'll be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Dr. Benedict Kessler, will now begin his presentation on translational proteomics and metabolomics. Benedict?
Thank you, John. I would like to thank Agilent for giving me the opportunity to present our work in this context. And what I would like to do is to show you a case study in a medical condition called ankylosing spondylitis and how a multi-omics approach allowed us to unravel some additional molecular features underlying this medical problem. Next slide. So in our group, we are studying the ubiquitin proteasome system, shown on the left, and which is actually the major pathway to regulate protein turnover and protein half-life in cells, and how this is connected to a variety of medical problems, human diseases, and how we apply mass spectrometry, mostly proteomics and metabolomic approaches to tackle these problems. Next slide. So one fundamental human disease that has underlying causes in aspects of the ubiquitin proteasome system is a specific version of arthritis called ankylosing spondylitis. And the major hallmark of that human disease is indicated in the left part of the slide that affects the human spine. And in the course of this arthritic disease, the ligaments and joints of the spine undergo chronic inflammation, and over time, through bone remodeling, new bone structures are forming within these joints and ligaments to then lead to fused discs and a complete rigidification of the spine, causing a lot of pain to the patient and also inability to move. So genome-wide association studies have revealed a number of genes of the ubiquitin proteasome pathway depicted on the right, that mutations thereof are actually associated with that disease. In particular, an MHC, or major histocompatibility class 1 molecule called HLA-B27, that as its function presents endogenous peptides on the cell surface, which are then recognized by T lymphocytes for immune recognition. And also a aminopeptidase called ERAP1, which is an endoplasmic reticulum located aminopeptidase that actually is involved in trimming these peptides to the right length, that are then presented on these MHC class 1 molecules on the surface of cells for immune recognition. So in one aspect of our work, we actually have solved the structure of this aminopeptidase, ERAP1. Next slide. Which is shown on this slide in two conformations. One is the closed conformation and one is an open conformation that allows to actually study how this aminopeptidase is actually trimming these peptides for loading to correct loading on MHC class 1 molecules. And we mapped current mutations into this structure that are actually associated with ankylosing spondylitis disease. And a critical mutation, the K528 mutation, we localized to the actual hinge region that actually opens and closes the enzyme to function correctly. Next slide. As a functional consequence of these mutations, it actually affects these enzymes how they can process peptides and trim them from the N-terminus, and this is shown in this slide. The experiment is actually an in vitro experiment with recombinant wild-type ERAP versus mutant ERAP enzyme, to which we fed peptide precursors, 13 mers, and let them digest them over time, and then measured their actual degradation products over time by LCMS using an Agilent QTOF instrument. And as you can see on the left of that slide, you can see disappearance of the initial 13-mer peptide, and then the reappearance of shorter degradation products over time. And if you then compare the kinetics seen with the wild-type ERAP1 as compared to the ERAP1 K528R mutant, you can see that they can degrade the original 13-mer peptide with equal kinetics, but then the actual further trimming of amino acids happens with different kinetics. In a sense, the mutant ERAP enzyme stalls after cleaving two amino acids and cannot further process it. So we see clear differences in the trimming of these peptides, and that must have functional consequences of what peptides are really presented by these MHC molecules and possible effects on modulating the immune recognition and probably causing part of the autoimmune properties that causes then the ankylosing spondylitis. So next slide. In another project in that context, we also wanted to know whether there is any other molecular features that can be associated with this disease, ankylosing spondylitis. And for this, a postdoc in my laboratory has delineated a proteomic and also a metabolomic workflow to analyze sera from patients with ankylosing spondylitis as compared to control patients. And for the proteomic workflow, it includes a depletion of Ig and albumin, and then a desalting step, and then a chromatographic separation and analysis by a high-end orbiter instrument. And for the metabolomic approach on the right side, it depicts a precipitation of proteins from sera, small molecular weight cutoff filtering, and then analysis by LC chip-cube MS on an Agilent QTOF instrument. So the first part, the proteomic part, if you go to the next slide, shows you a list of differentially expressed proteins that we found in sera from control as compared to AF-derived patients. And from about 600 proteins that we could identify using that approach, there are about 20 proteins that are significantly differentially abundant between the two cohorts. And most of these proteins, as you can see here, correspond to or belong to the pathway of inflammation, but also what we call the acute phase response. Interestingly, a few components that we identified, including serum amyloid P component, for example, does not show the trend that we usually see in inflammatory response, indicating that although there is chronic inflammation markers in there, there is something peculiar about the acute phase response that underlies the etiology of ankylosing spondylitis. At the same time, we also looked at the metabolite or the metabolome, what we could resolve by LCMS in sera of these patients as compared to controls. and we were able to detect up to 7,000 molecular features in these LCMS runs when we looked at processed sera from these patients as controls, and we were able to identify molecular features that were actually differentially present. The next slide shows you an example of that, where you can see the isotopic profile in a 3D representation of a particular molecular feature that is upregulated in sera from AS in the green display as compared to the healthy controls in the orange display. And these trends are quite consistent through the different patient cohorts, as you can see in the lower left panel. We have then tried to subdivide the ankylosing spondylitis patient cohort into subpopulations depending on disease severity, and that is shown explicitly in the next slide. Using the BAS Ankylosing Spondylitis Disease Activity Index, or BASDI index, and if you use a principal component analysis to cluster or to subdivide ankylosing spondylitis subjects into disease severity, we could see a remarkable correlation of subgrouping using PCA of the different subgroups. So, for instance, you can see the healthy control shown here in pink groups together, and the subsequent groups with increasing BASDI index then represent separate subgroups that move to the right and then to the top. So there is a clear trend in their molecular feature, or how their molecular features in their sera actually changes as a function of disease severity. So next slide. The question, of course, is what are these molecular features? This, of course, is a big challenge in the field of metabolomics to then identify these components. Nevertheless, we were able to identify one of these components that is actually present in sera of healthy controls. You can see that in a 3D representation in the upper left. That completely disappears or is present at lower levels in sera of ankylosing spondylitis subjects. And in panel B, upper right, you see the dot plots indicating the statistics for this differential abundance. And we were able to use targeted MSMS to obtain an MSMS spectrum of sufficient quality to actually not only identify the actual molecule candidate, but also increase the confidence of identity by actually assigning fragment ions to subparts of the molecule. And that gave us the identification of this peak to be a vitamin 3 derivative, 2623 peroxylactone derivative of vitamin 3. Next slide. We were able to confirm the identity of that particular metabolite by comparing the theoretically expected isotopic profile of molecules of that with that formula indicated in red as compared to the experimentally measured isotopic peak distribution that we have measured for that particular parent ion mass, either as a singly charged or as a monomer in the left panel, also observed as a dimer in the middle panel, and to the right also observed as a trimer. And only the isotope distribution of the measured molecule fitted exactly to one particular molecular formula, the top one indicated in red, that actually is exactly consistent with vitamin D3 molecular structure that I've shown before, providing further evidence that the identification that we made is correct. Next slide. We then, once we identified this vitamin 3 derivative, we then went back to the proteomics data set and noted that we actually identified a vitamin D binding protein that was also differentially present in sera of control versus ankylosing spondylitis patients, and that the possible correlation between the differential abundance of the vitamin D binding protein indicated on the x-axis as compared to the differential abundance of the vitamin D3 analog indicated on the y-axis showed us that in control sera, the correlation seems to be rather different than in sera of AF patients, indicated in black, meaning that somehow there are components of the vitamin D3 metabolic pathway that seem to be dysregulated in sera from ankylosing spondylitis patients as compared to control subjects. Next slide. So what does that particular vitamin D3 derivative do? The form that we actually found or identified is not the active version of vitamin D3 that actually is responsible for a number of physiological effects, not only bone resorption and remodeling, but also more recently vitamin D has been implicated in diverse immune effects, and that's on the next slide. So here you see a schematic representation of the vitamin D3 biosynthetic pathway to the left, and some of their physiological functions summarized in a schematic presentation on the right. And the derivative that we identified is actually an inactivated derivative of the active component of vitamin D3 that we've seen accumulating in our sample cohort. So if you go to the next slide, this shows a more schematic representation of the vitamin D metabolism, and you can see that the vitamin D binding protein, indicated with the red circles, is actually implicated in transport of vitamin D3 either into hepatocytes or further downstream also for another aspect of the pathway. The actual vitamin D3 derivative that we identified is a byproduct of the kidney and actually gets secreted out and gets back into the circulation. And the biological activity of that derivative is not known. It's probably an inactive one that accumulates. But you can also see from that particular representative scheme that there are lots of other components that we haven't really been able to look at that would tell us probably a more complete story. So they are an integrative approach. In this case here, a metabolomic versus proteomic approach could shed us or could provide us much more information on what's really happening within the context of disease. So if you go to the next slide, we are still quite far away from sort of having a more global view of these metabolic pathways in a sense, but I think our story or our findings could presented a initial start or initial insight into something in the vitamin D metabolism that is not or dysregulated in the context of this arthritis disease. So if you go to the next slide, these are the conclusions. So we have used combined proteomics and metabolomics approach to address molecular mechanisms in a medical problem, in this case here ankylosing spondylitis. And these approaches are actually not without their limitations, so there are little useful applications that have yet been transferred to the clinic, but there is a big promise that combining information that comes from different omics approaches could provide a huge step in understanding and advancing knowledge behind such medical problems as ankylosing spondylitis. The first part of the talk showed the effect of ERAP1 aminopeptidase polymorphisms that are associated with AS, that this affects N-terminal trimming of antigenic peptide epitopes. Therefore, a manipulation of this aminopeptidase by small molecule inhibitors might provide a way to for treatment of ankylosing spondylitis. And in the second part of the talk, I showed you some evidence that there is an altered vitamin D homeostasis observed in patients with ankylosing spondylitis. This actually does make sense because of this bone fusion that we see in the spine must indicate a dysregulation of bone resorption and bone remodeling that is heavily dependent on vitamin D, therefore provides another entry point for therapeutic strategies to interfere with this aspect of ankylosing spondylitis. And at the end, I would say thank you for the people on the next slide the people who actually contributed to that work. So most of the work that I presented today has been performed by Dr. Roman Fisher, top right, who is a postdoc in my lab. And some of the bioinformatics was done with Dr. David Trudgeon, who is the third one on the left, who helped us out with the data analysis, which in my view is a key feature required if we want to start to understand integrated multi-omics data sets in the future. Thank you very much for listening, and I'll pass my work back again to John. Thank you.
That was a wonderful presentation, Benedict. Thank you very much. I think the overview on proteomics and metabolomics workflows and the accompanying discussion on ankylosing spondylitis were particularly intriguing, so we really appreciate that. Before we proceed, let me remind everyone of our Q&A segment that comes right after the panelists made their presentations. Please type your questions into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist, Dr. Akhilesh Pandey, is ready to talk about the benefits of an integrated transcriptomic and proteomic analysis in relation to the pharmacodynamic study of an anti-cancer drug. Akhilesh, the floor is yours.
Thank you, John. Today I'll talk about our integrated transcriptomic and proteomic analysis to determine the pharmacodynamic action of sulforaphane, which is the chemopreventive agent. Next slide. Many of you might know that there are two FDA-approved agents for breast cancer chemoprevention. These are tamoxifen and raloxifene. Both of these agents target the estrogen receptor. This means that estrogen receptor-negative breast cancers, which are often more aggressive, have no approved chemoprevention options. We need additional molecular targets for chemoprevention of breast cancer. Next slide. Sulforaphane, which is an isothiocyanate, most famously isolated from broccoli sprouts, is a breast cancer chemopreventive agent that has been studied in cell lines as well as animal models, including breast cancer models. Next slide. This slide shows the pathway that is known to be affected by sulforaphane treatment. The central players in this pathway are proteins called KEAP1 and NRF2. Sulforaphane binds to cysteine thiols in KEAP1, and this prevents KEAP1 from interacting with NRF2 and causing degradation of NRF2 through the proteasome degradation pathway. When sulforaphane is bound to KEAP1, NRF2 can then go to the nucleus and bind to regions upstream of antioxidant class of genes that have antioxidant response elements. And the slide shows some of the most studied members of this family, such as NQO1 or NADPH, quinone oxidoreductase, HMOX1, heme oxygenase 1, and others. These genes are involved in detoxification mechanisms in the cell and ultimately lead to chemoprevention. Next slide. Our hypothesis is that sulforaphane prevents breast cancer in humans in part through modulation of gene and protein expression, and that this response leads to altered regulation of pathways that protect the mammary glands from both endogenous and exogenous carcinogens. Next slide. The pipeline that we are following is to essentially find biomarkers that would provide a readout of the action of sulforaphane. So we are carrying out an unbiased screen, which is for discovery of these candidate biomarkers, and then we move on in a targeted fashion to carry out an initial validation to confirm differential abundance of candidate biomarkers, and then on to validating these candidate biomarkers in actual clinical samples. Next slide. We have decided to use the MCF10A breast epithelial cells as our system to look at the effect of sulforaphane. These MCF10A breast epithelial cells are non-tumorigenic and are estrogen receptor negative cells. As I mentioned earlier, this is the subtype of breast cancers for which we do not have any chemoprevention agents available today. And initially, we set out to optimize the dose, time, and methodology to carry out our transcriptomic and proteomic analysis. Next slide. This slide shows how we are able to do a dose response to look at the classic readout of the NRF2 KEAP1 pathway, which is NQ01 transcript, that is the RNA, as well as the biochemical activity of NQ01. In this case, measured at 24 hours for transcripts and 48 hours for the activity of the enzyme. This allowed us to determine the time frame and the dose to be used for subsequent experiments. Next slide. This slide shows the overall strategy of our experiments. Here, our goal was to compare the effect of pharmacologic intervention with genetic intervention. The pharmacologic intervention was treatment with either sulforaphane or a vehicle, and the genetic intervention was an siRNA that was used to knock down KEAP1. And here our goal was to look at the effects that were induced by sulforaphane and to compare them to what we saw when the major pathway that is known to be downstream of sulforaphane was targeted, that is the KEAP1-NRF2 pathway. As you can see on the slide, we did both transcriptomic and proteomic experiments. The transcriptomic experiments were done at 24-hour time point using Agilent's whole genome microarrays. The quantitative proteomic experiments were done using the SILAC method, or stable isotope labeling by amino acids in cell culture, at a 48-hour time point. And here we use LC-MS-MS analysis on Agilent's QTOF mass spectrometer. Next slide. This slide shows hierarchical clustering of some of the differentially expressed genes from the KEAP1 knockdown experiment on the left and the sulforaphane-treated experiment on the right. The panel on the left shows 4 biological replicates for control siRNA and 4 biological replicates of the KEAP1 knockdown siRNA. What you can see is that there are a number of genes that are upregulated when KEAP1 is knocked down. And if you pay some attention to some of the gene symbols from the panel on the left and then move over and look to the right, you see many of these names are repeated because some of the genes are common between KEAP1 knockdown and sulforaphane treatment. And in the next slide, this can be visualized much more easily, which shows a subset of what we saw on the previous slide. We can easily look at the overlap between the KEAP1 and the sulforaphane treatment conditions. Next slide. This slide shows a Venn diagram of the transcriptomic changes that we observed in these experiments using the fold cutoffs indicated on the slide. What can be easily appreciated is that the number of transcriptomic changes induced by sulforaphane is far numerous than KEAP1 knockdown induced changes. But there is a substantial overlap of 879 genes across the 2 conditions. Next slide. This slide shows the workflow of the proteomic analysis that we carried out. If you look at the panel on the left, it shows the cells were either treated by a vehicle or by sulforaphane. And those that were treated by a vehicle were grown in a light medium, and those that were treated by sulforaphane were grown in a heavy isotope-containing medium. In this case, the isotope was contained in the heavy amino acids. After the cells were labeled and treated as shown, the cell lysates were mixed, we ran a gel, and carried out in-gel digestion of all of the bands on the gel. followed by LC-MS analysis on an accurate mass Q-TOF mass spectrometer. We carried out protein identification quantitation using the SpectrumMill software. The panel on the right shows similar experiments of a control siRNA versus keep-one siRNA-treated cells. Next slide. The two panels shown here summarize the data of fold changes observed in the sulforaphane treatment experiment and the keep-one siRNA knockdown experiment. As you can see, that the majority of the proteins under both conditions do not actually change, but a small minority of proteins show a difference in abundance. We see both evidence of up and down-regulated proteins in both conditions. Next slide. This Venn diagram shows the overlap at the protein level this time of those proteins that change more than 1.5-fold in the two different experiments. Again, as you can see, that there is an overlap between the two experiments, but then there are also distinct changes that were identified in one condition but not the other. Next slide. This table summarizes some of the changes that we saw at the transcriptomic and the proteomic level. I would say this is the way we used to analyze the data, where one had to pour through individual entries and then go across different columns showing different values. But this has changed dramatically with improvements in software that allow us to integrate data and also to analyze and visualize data better, as is shown on the next slide. This is a multi-omic pathway analysis that we carried out using the latest version of GeneSpring, that is version 12. And as you can see in the panel on the left, what you see highlighted in blue is one of the pathways called biotransformation. These pathways have been imported by us from wiki pathways into GeneSpring. And you can see that this was one of the significantly enriched pathways that was found to be common between the sulforaphane and the keep-one knockout experiments. And the arrow points to the pathway image that can be visualized within this program. Because this may be harder to see, the next slide shows a zoom-in view of the overlay of the transcriptomic data onto this biotransformation pathway. And the areas that are circled indicate those genes where we saw an overlap between the keep-one knockdown experiment and the sulforaphane treatment experiment. Next slide. This slide now shows a different view of the same pathway. Here, instead of overlaying the transcriptomic data onto the pathway, we are overlaying the proteomic data from the keep-one knockdown and the sulforaphane treated experiment. Next slide. This is another section of what we found in this case using the multi-omic pathway analysis resource within GeneSpring. The arrow shows on the right a zoomed out view of the keep-one NRF2 pathway, and the next slide shows how we've been able to integrate transcriptomic data related to the keep-one NRF2 pathway. You have to remember that we have the flexibility to import these pathway data sets from a variety of data resources, and it's also possible for you to create your own pathway and then upload it in the software. So here you can visualize the transcriptome overlaid onto the keep-one NRF2 pathway, where you can easily visualize what is changing or what is not changing in the two different experiments. The legend is at the bottom right. And the next slide shows a view of what we found at the proteomic level in this pathway. The next slide shows how one can interrogate these genes and proteins that one finds within this by using additional functionality within the software. This is a view of the GeneSpring Genome Browser, where if you wanted to look at the genomic view and the transcript view of any of the transcripts, you can enable that by using the Genome Browser, and this allows you to be able to look at transcript structures, for instance, and to see how that might impact your biological experiment. The next slide. This slide shows a Western blot-based validation of the data that we got from our SciLac experiments. Here we have used not only MCF10A cells, but also MCF12A cells that is also commonly used in breast and breast cancer research. What we are easily able to validate using commercially available antibodies is that these expression changes that we observed by quantitative mass spectrometry, we can also see by Western blotting. However, one can appreciate that here we are lucky in that commercially available antibodies that work in Western blots are available, and that may not be the case with the large majority of proteins one finds from mass spectrometry experiments. The next slide summarizes some of the candidate biomarkers of sulforaphane activity that we identified from our series of experiments. The next slide shows how we have initiated efforts to translating our results from discovery experiments onto experiments that would be more appropriate in a clinical setting. Often in a clinical setting, one takes a biopsy and the tissue is then converted into paraffin-embedded sections. To mimic that situation, what we have done is we took the cell line that we are working with, MCF10A cells, we treated them with sulforaphane or knockdown using the siRNA against KEAP1, and prepared cell pellet blocks that would mimic a tissue block. And then we use these antibodies that are available against individual proteins to see if we are able to visualize the biomarkers that we identified from our transcriptomic and proteomic experiments. And this slide nicely illustrates how one can easily appreciate the upregulation of this gene product, AKR1C3, using immunohistochemistry. The next slide shows how our real goal was to try to move into a clinical setting to carry out our final validation in mammary tissues. Next slide. For this, we turned to mammary organoid cultures, which are mammary ductal structures that can be maintained in cell culture, and in this case, we obtained them from reduction mammoplasty surgery. Next slide. When we treated organoids with vehicle or sulforaphane and prepared protein extracts, we are able to carry out the same kind of Western blotting experiments that I showed earlier, where we are able to monitor changes in protein level. And again, we saw a nice correlation of the data that we obtained from cell line experiments now in organoid cultures, taking us one step closer to clinical translation for sulforaphane action. Next slide. In summary, what we have found through this integrated transcriptomic and proteomic analysis is that there was a strong correlation in the mRNA and protein changes across the sulforaphane treatment and KEAP1 knockdown experiments. We also identified common pathways for sulforaphane treatment and KEAP1 knockdown, and these were biotransformation, glutathione metabolism, NADH and NADPH regeneration, among many others. Next slide. The most exciting part of our findings was that we were able to identify many candidate biomarkers that had not previously been identified, and we identified them through this overlay of transcriptomic and proteomic evidence, where we increase our chances of success through this integration. And we also were able to test some of these not only in the cell line system, but also in a more clinically relevant situation of using organoids from reduction mammoplasty. We are now carrying out experiments to do experiments using breast tissue biopsy from clinical trials that are ongoing. Next slide. This was truly an interdisciplinary study, and several people participated in this project. The main person who carried out the experiments was Abna Agyeman, and she was assisted by Ragotma Chirkadi, Patrick Shaw, and Arivusudha Marimuthu. Others involved were Kalla Vishwanathan, Tom Kensler, and Nancy Davidson. And I would also like to thank scientists at the Institute of Bioinformatics in Bangalore, India. And back to you, John.
And thank you, Akhilesh. I want to extend my appreciation to you and to the entire panel for your excellent presentations. Looks like you all hit a resonant chord in our audience as we received a number of pertinent questions from them. So let's get started with our Q&A segment. And the first question will be for Q. Rhee. Q, what does metabolomics measure? Whether a drug acts on known metabolic pathways or whether the drug itself is metabolized by analyzing its breakdown products?
Thanks, John. So actually, it measures both, and that's part of the beauty of it is that it simply surveys the chemical composition of cells, and then one sees not only drugs but the effects of the drugs, so both the PK and the PD, and so the data set once collected can be used in both ways depending on how one filters the data sets.
And Q, we have another question. How easy was it to integrate wiki pathways with GeneSpring?
Right. So that was actually a key feature that we actually was essential in the development of the software. That by design it's meant to interface with wiki pathways so that anybody could have the flexibility to be able to use or curate their own custom pathways and not be constrained to other people's databases or textbook diagrams, because a lot of what omics technologies is really doing is kind of experimentally redefining what we assume to be true, but experimentally often find to be different, and to be able to respond to that. So by design, the software was meant to integrate with wiki pathways.
Well, thank you. And Akhilesh Pandey, the questioner also directed that particular question to you. How easy was it to integrate wiki pathways with GeneSpring?
Yes, John. As Q pointed out, it was very easy and it was very seamless integration, and we are actually very excited about that feature. And also, as Q pointed out, that it actually enables researchers to upload their own pathway data, and we are an excellent example of that, because the Institute of Bioinformatics in Bangalore that I also direct, we have created a very nice web-based resource of signaling pathways called NetPath. And this resource has been published standalone, but many of the pathways have been exported and are an integral part of wiki pathways, and we are able to capture all of that from wiki pathways and use it for the analysis of the data that I showed during my presentation.
Okay, Akhilesh, and someone wants to know, what advice do you have for a lab initiating transcriptomics approaches in mammalian cell culture systems, especially for data analysis and informatics?
Yeah, that's a very good question, and I think one has to point out that transcriptomic approaches, especially by gene expression microarrays, they have become very standard now, and let's say 5 to 10 years ago, people used to argue about many of the different statistical and other approaches that people take. But what has really happened is commercial software such as Agilent's GeneSpring has made it very easy for people to not focus on many of the issues that people used to argue about, but instead to focus on the biology. And these types of software have made it very easy at the graduate student or postdoc level folks to be able to upload their data and follow a very easy protocol to be able to analyze the data still in a statistically robust way, but they don't need to know any programming or anything like that, and then be able to analyze down to the pathway level about what the data is saying. And as was pointed out by the other speakers also, that this software also allows you to integrate data from the other omes. So were you to go from transcriptomic to proteomic to metabolomic type of data sets, you can also integrate that all within the one piece of software. So I would say that if one is not so inclined to learn programming, for which there are many other tools out there, one can these days try to use commercial software, which makes it easy so that one can get back down to the biology quickly.
And Akhilesh, a question. What's the correlation between mRNA and protein abundance?
Yes, that's also a very interesting question, and there are certain groups that have reported their findings from many different systems, and I think it's fair to say that although people have found a correlation between a transcript and protein, earlier it was assumed that there's a very tight correlation, and many studies have now shown that the correlation coefficients can range from, say, 0.4 to 0.5 to 0.7. So what that basically tells you is that although many of the genes are correlated at the mRNA and protein level in terms of abundance, there is a very substantial chunk of genes and proteins that actually may not correlate, and that's why I think it's important if one has the chance to be measuring both mRNA and protein precisely because of that reason.
And Akhilesh, how comprehensively can one do a proteomic analysis today?
Until now, people were resorting to transcriptomic studies because they were high throughput, and one could essentially look at the entire transcriptome on a gene expression array, and that was not true for proteins because of the mass spectrometry methodologies and the limitations with sampling the entire dynamic range. People were not able to sample most of the proteins in samples. But today, I think it's rapidly changing, mostly because of sensitivity and advances in instrumentation, but also because one should do deep fractionation to be able to sample the proteome, and the net effect being, for example, for the human proteome, if one does a careful analysis, out of 20,000 odd protein coding genes, one is able to sample, say, 6,000 to 10,000 of them in proteins in a single series of experiments and obtain quantitative data on them.
Well, thank you, Akhilesh. And Benedict Kessler, we have some questions for you. What software did you use to analyze your proteomics and metabolomics data?
So in our case, we were not using GeneSpring directly, but for the proteomic data, we used our own proteomic analysis pipeline, which is a version of the TPP from the ISB, combined, so different search engines combined, and then iProfit for further validation. And the metabolomic data sets were analyzed using Mass Profile Professional from Agilent, but also LC Progenesis software combined. And so far, the integration into pathways, we also used wiki pathways as a sort of a background to map our data on it. We greatly look forward to try to integrate GeneSpring, latest version of that, into these workflows in the future.
And Benedict, what is the integrative aspect within the analysis of your study on ankylosing spondylitis?
Initially, we didn't plan it to integrate anything at all. It was the biology, actually, and the results that we obtained that prompted us to try to do that. And this is the how we gain confidence that in this particular disease, this ankylosing spondylitis, which is this arthritis disease, that there's something wrong with vitamin D, with the vitamin D metabolic pathways. And by looking at each individual one, I think we were not convinced that there is something real there, but I think the combination of looking at the different data sets convinced us that, yes, there must be something that's not correct there. And that provides the framework to dig deeper into the data in that direction.
And Benedict, what additional specific upstream to metabolite analysis tools and methodologies do you believe would need to be developed to overcome the analytical incomplete coverage?
So I think the analysis of metabolites is probably one of the most difficult one to cover in a comprehensive manner, and this is because small molecules and metabolites, their physicochemical properties vary dramatically. So it's not like peptides that all have physicochemical properties to be able to be resolved by reverse phase LC. In the case of metabolites, you have lipids, you have small polar compounds, you have aliphatic compounds, and each class requires sort of a different chromatography or analytical approach to isolate. And some of them that have been used, the one I presented is based on liquid chromatography, but we are fully aware that we only capture a subset of what you would call the metabolome. This certainly should be complemented with gas chromatography or GCMS and also NMR studies. But I think another promising technology that has been used for the analysis of metabolites is capillary electrophoresis MS, so CEMS. And it's only by combining all these that we really can increase the coverage and the measurement of as many metabolites as possible. But certainly this is a key challenge in that area that will occupy us for the years to come, I would say.
Thank you, Benedict. And a question came in for Q. Can we identify a macrophage transcriptome signature specific for ingested bacterial species?
Yeah, so actually that was something that was done by Eric Lander and Rick Young almost 10 years ago now, I think, with whatever that generation of microarray technology was at the time. And even at those kind of early days of microarray technology, they reported that it was clear that you could, that there were common patterns across bacteria as well as distinguishing features, but I'm not aware that anyone's really gone back with modern technologies to try to improve this and translate this into something diagnostically or clinically useful.
Thank you, Q. And Akhilesh, that same question for you. Can we identify a macrophage transcriptome signature specific for ingested bacterial species?
Yes, I think it's a very interesting question, and I would go on to say that now that we have proteomic technologies that are actually very advanced, that the SciLac method can easily be adapted to a question like that, where you're looking at what are the proteins that are changing in response to the transcriptomic signature, then one can identify. And what I would venture to say is that these kinds of experiments, I think, are going to take off in the very near future, because until now, people have been not focusing so much on the host pathogen response, but now we have all of the technologies falling in place that we can really use all of these tools in our arsenal to look at not just transcriptomes, but also proteomes in response to situations such as bacterial infection.
And Akhilesh, another question. Have you seen any different responses between the cell line and the primary tumor? Have you seen the profiles of mRNA and proteins with or without the estrogen receptor?
So the first part of that question, I think if there's any different response between cell line and primary tumor, I think I would like to answer that in a generic fashion. And I think people in the community often ask that question because, cell lines are cultured in vitro, and people are always worried about what exactly happens and if that is reflective of what happens in tumors. And surely there is a lot of contribution by the microenvironment, but if someone is asking questions such as biomarkers, then we have done many types of studies, not the one that I talked about, where we have been looking at biomarkers, where we do our initial studies in cell lines and then go back to tumors. What is striking, at least to us, is that we often find what we initially discover in cell lines to be true in tumors. most people, including ours, we don't do side-by-side comparison of tumors versus cell lines because when one is analyzing tumors, it's really a mixture of cells. And talking about this comparison of profiling mRNA or protein with or without estrogen receptor, I think that has to be done in a very carefully controlled way. And I think simply taking, say, tumor from a patient where the tumor is estrogen receptor positive versus another patient's tumor which is estrogen receptor negative is unfortunately not a great experiment because there are many other differences across the two patients. So here now, to answer that question, one is forced to go into a cell line. And we have begun a nice series of experiments in collaboration with several other investigators here at Hopkins, where we develop isogenic cell lines, where essentially you have a cell line that expresses a given protein, and you have a sibling of that cell line derived from that cell line, which has everything except one gene. We have not done that for estrogen receptor, but we are doing that for a variety of other molecules that are important in cancer, and I would imagine that one can extend that to estrogen receptor also, and we are getting very nice answers out of those isogenic systems where everything is identical except one protein. So it's a very good question, and again, I would say that these kinds of very nice questions would be asked by more and more people in the cancer and also other communities.
Nicholas, thank you so much for that detailed response. With that response, though, we're going to need to end the webinar because, unfortunately, we run out of time. Please note that the webinar will be archived for 6 months on our website, www.GENNews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. Thanks again to the panel for the outstanding presentations, and I want to say thank you to our audience for your attention and for your thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, Agilent Technologies, who made this event possible. Scientists at Agilent are on board with the concept that integrated multi-omics techniques that utilize the wealth of prior biological knowledge constitute a powerful and important tool for biological researchers. Agilent's integrated biology software simplifies this type of approach through the use of its Pathway Architect module. Agilent officials understand the challenge and are committed to partner with scientists to move this research field forward. Shortly, we'll be sending you a survey, and we would appreciate your feedback on this webinar. Please look out for it and kindly give us your thoughts, as this will help us to continue to bring you topical and timely webinars in the future. Thank you.